Everyone watching this lecture was once a single cell, and all the cells in a multicellular organism, like you or me, have arisen by division from a single fertilized egg, and therefore they all have the same DNA as that egg. Division of a fertilized egg produces, in the case of humans, well over 1 trillion cells by the time a baby is born. Yet we also know that a baby is not a giant ball of 1 trillion identical cells, but has the many different kinds of cells that make up tissues like skin and muscle and bone and nerves. How did cells that have identical DNA turn out so different? The answer lies in gene expression, which is the process by which the information in DNA is used. Although all the cells in a baby have the same DNA, each different cell type uses a different subset of the genes in that DNA to direct the synthesis of a distinctive set of RNAs and proteins. Cells can respond to change by increasing or decreasing the activity of particular proteins, but they can also respond by altering the proteins that are made at all. Here, cells are responding to change by regulating gene expression. That is, we are now adding another level of regulation of proteins beyond the control of their activities. Regulation of gene expression determines which genes are copied into RNA and translated to make proteins. Variations in the proteins that are made also explain why, in multicellular organisms like ourselves, there are many different kinds of specialized cells doing different things despite having the very same DNA. In the lectures on transcription and translation, you got to learn about the steps by which RNA and proteins are made. Now we will examine the controls that determine which genes will be expressed at a given time, where in the body, and to what extent. Cells have an amazing array of mechanisms to regulate gene expression. You will learn how a variety of mechanisms at various stages in gene expression regulate the kinds and levels of proteins available to the cell. We'll examine some epigenetic mechanisms that act on the information in DNA to control how it is used. You'll learn how the food you eat can affect which genes are expressed and turn you into royalty, if you're a honeybee, that is. And you'll take a look at how recently discovered regulatory RNAs are opening new avenues in treating disease. Gene expression is a multi-step process which provides many points at which cells can regulate the production of proteins. You've already learned that transcription is the first step in gene expression, so let's begin with how transcription can be regulated. And for this, let's start with bacteria, not only because they're simpler, but also because bacterial gene expression is what drives the workings of our gut microbiome. Bacteria are marvels of efficiency from a biochemical standpoint. Their genomes are compact. Genes are often clustered in convenient groups in bacteria, such that genes that need to be expressed at the same time are next to each other. This physical proximity allows them to be controlled as a single unit through a single promoter. Groups of genes operated on together like this are called operons. In 1965, the first operon was discovered in a group of bacterial genes called the lac operon, so named because they control and metabolize the sugar lactose. There are 3 genes in the lac operon called LAC-Z, LAC-Y, and LAC-A, and they're under the control of just one lac promoter. Let's take a look at how the lac operon is regulated. As you know, lactose is the sugar in milk that some of us are unable to digest. Lactose isn't E. coli's favorite food either. It avoids using lactose if its preferred sugar of glucose is available. But if it runs out of glucose and finds lactose available, well, a hungry bacterium has got to do what it's got to do, right? It switches on the lac operon so it has the proteins needed to take up and use lactose. So how do E. coli cells switch the lac genes on as needed? Managing the on-off switch is the work of 2 proteins, one a repressor that prevents transcription when there's no lactose, the other an activator that stimulates transcription when glucose is low. Which do you suppose is the default setting for bacteria? On for using lactose or off? Well, because bacteria don't normally use lactose, the default state for the lac operon is off. What keeps it off is the lac repressor protein, which is usually bound to a small region of DNA called the operator, right in front of the -10 sequence of the lac promoter. The repressor is thus sitting squarely in the path of any RNA polymerase that might bind at the promoter. In fact, the repressor is hogging a bit of the space that RNA polymerase would occupy when it was bound at that promoter. Like a car blocking a driveway, the repressor prevents the polymerase from binding to its promoter. It can't get in the driveway except if there is lactose present. If lactose is present, a small amount of it enters the bacterial cell, where the lactose gets converted into an isomer called allolactose. It is the allolactose, not lactose, that binds the repressor, loosening its grip on the operator. When this happens, the repressor comes off the DNA. In biochemical lingo, the allolactose is called an inducer. You can think of the allolactose as the driver of the car that was parked in the way. When the driver gets in the car can move out of the way. Okay, this means there isn't a barrier, but transcription still isn't going to happen unless RNA polymerase binds at the promoter to transcribe the genes. It turns out that RNA polymerase can be lured to the lac promoter by a catabolite activator protein called CAP, C-A-P, which binds near the promoter. Upstream from the promoter on the DNA sequence, there's a spot called the CAP binding site. When glucose levels are low, CAP binds upstream of the promoter. That recruits the RNA polymerase to come bind to the promoter and begin transcription. What tells CAP that glucose levels are low? Well, when glucose levels are low, an enzyme called adenylate cyclase is activated. This enzyme makes the molecule cyclic AMP or CAMP. When there's lots of cyclic AMP, it binds to CAP. This leads to a conformational change in CAP that allows it to bind to the CAP binding site. Okay, so CAP is bound near the promoter. When glucose is low and CAP can recruit RNA polymerase to come bind the adjacent lac promoter, this increases the amount of transcription that occurs. So the repressor is moved when lactose is present, and the RNA polymerase is recruited to the promoter when glucose is depleted. With both of these conditions, the lac genes can be transcribed at high levels and the proteins needed to break down lactose are made. Okay, great. Genes are turned on to make use of lactose. What happens when the lactose gets all used up? Well, when lactose levels drop, there's not much allolactose around. And without allolactose to ease its grip on the DNA, the repressor goes right back to sitting on the operator, and the expression of the lac genes is shut off once again. Pretty neat, huh? This example illustrates a common theme in the regulation of transcription. The binding of proteins to specific DNA sequences can increase or decrease transcription from a promoter. I marvel at E. coli, as everybody knows. They have a way of managing their many sugar woes. When they do not see lactose, its operon's not expressed, because the lac repressor keeps everything repressed. But when lactose appears in cells, a good researcher finds the lac repressor snatches it, no DNA it binds. Then RNA polymerase sees open operator, but doesn't really do too much until the need is greater. This happens when the cell has got no other energy, and then it starts to make a lot of cyclic AMP, which binds transcription factor and the DNA it wraps. I think you might have heard of it, a protein known as CAP. When this occurs, a CAP binds to its cleverly named CAP site. Then RNA polymerase makes RNA all night. Now let's see what happens in eukaryotic cells, including our own. The binding of proteins to specific DNA sequences is one of the mechanisms of regulation in eukaryotes, though it works a bit different than in bacteria. In eukaryotes, genes commonly contain two groups of sequences important for transcription. First, as you've already seen, are promoters. Their job is to initiate transcription by assembling the basal transcription complex. Additionally, in contrast to bacteria, eukaryotic cells also have regulatory sequences affecting transcription that can be thousands of base pairs away from the gene. Some of these are enhancers that work to increase transcription of a gene, and others are silencers that reduce transcription. Let's take a look at these forms of remote control. First, the sequences must be bound by specific proteins that either activate or repress transcription. This is a familiar theme, similar to how transcription in bacteria can increase or decrease. But for eukaryotes, the enhancer or silencer sequences are far, far away on the DNA molecule from the gene that they control. What makes it possible for a regulatory sequence to influence what happens at a distant promoter? An enhancer or silencer with its bound protein can be brought close to the promoter of the gene being transcribed by looping of the DNA strand. That is, when transcriptional activators bind at these distant sites, the DNA strand bends. This allows the proteins bound at enhancers to come close to the transcription initiation sites, where the activator proteins can interact with the basal transcription complex. One effect of this interaction is to help in recruiting proteins necessary for transcription to the promoter. More of these proteins means the transcription initiation complex is made more efficiently and more frequently. Another thing that transcriptional activators bound at enhancers can do is recruit enzymes to help open up the chromatin structure. The chromosomes of eukaryotes have DNA packaged up with proteins to form a complex called chromatin. This means that when a region of DNA has to be transcribed, it must be separated from the proteins temporarily to allow the RNA polymerase to transcribe it. And how do the enzymes open up the chromatin? Remember that DNA in our cells is wound around histone cores to make structures called nucleosomes. Histones, being proteins, are chains of amino acids that fold to take on three-dimensional shapes. But one end of the amino acid chain making up each histone hangs out of the bead-like nucleosome. These tails are targets for the enzymes which add one or more small tag molecules on to specific amino acid side chains. For example, an acetyl group may be added to one amino acid side chain and a methyl group added to another. Or an acetyl group may be added to one while a phosphate is added to another. Depending on the particular combination of these chemical tags, the chromatin in that region will be loosened up to allow transcription or packed tightly to silence the expression of genes in that part of DNA. One enzyme targeting the tails of amino acids is called histone acetyl transferase, or HAT. It transfers an acetyl group to residues in the tails of histones. Acetylation of histones loosens the interaction between the histones and the DNA in nucleosomes, making the DNA more readily accessible for transcription. The opposite effect may be achieved if the enzymes recruited are histone deacetylases, or HDACs, which remove acetyl groups from the tails of the histones. Lack of acetyl groups leads to tighter packing of the chromatin. Tighter packing naturally would silence transcription. One of the most extreme examples of shutting off gene expression is the long-term silencing of an entire chromosome by tight packaging of the chromatin. The chromosome in question is one of the two X chromosomes in human females. As you know, human males have only one X chromosome plus a Y chromosome, while females have two copies of the X chromosome. This means that females have twice as many copies of the genes on the X as males. Now, to ensure equality, one of the X chromosomes is shut down by packing it up more or less permanently into very tightly condensed chromatin. This inactive X shows up in female cells as a small blob called the Barr body, after the scientist who first reported it. Before better tests were devised, the presence of Barr bodies was used to check that competitors in the women's Olympic events were actually female. Okay, so it's understandable that transcription in eukaryotes can be regulated by opening up chromatin or tightly packing it up. But there's more. It turns out there are enzymes that can modify the DNA itself, adding methyl groups at specific places. In our DNA, for example, enzymes called DNA methyltransferases can add methyl groups onto cytosines. Not all cytosines, mind you, but cytosines that are next to guanines in a single strand. That is, where you would see a C followed by G in the DNA. Now, not every C in a CG sequence gets methylated, but what we see is that methylation of DNA near transcription start sites is accompanied by silencing of the gene. Transcription gets reduced. By contrast, demethylation goes hand in hand with increased expression. Why? We don't know for sure, but it may be that methylation of the DNA at promoter regions prevents transcription factors from binding there. Thus, although DNA is the information storage molecule, mechanisms like histone modification and DNA methylation control what cells do with the DNA information. On its own, DNA is like the songs you may have on your phone or in a library. But what songs are in a playlist and what order these songs are played in is determined by the histone modifications and DNA methylation patterns. This also explains how all the cells in a baby's body arise from a single fertilized egg, but turn out so differently by the time the baby is born. The trillions of cells in your body all have the same DNA, but they have differentiated into a variety of different cell types, each performing a specialized function. In other words, all the cells have the same songs, but their playlists and the order in which the songs are played are different. This sort of playlist control over what cells do, which acts over and above the information in DNA sequence, is what we call epigenetic control. For example, the overall locations of the chromosomes in relation to one another may vary by type of cell. Such variations in what are called chromosome territories may also play a role in how the genes for each type of cell are expressed. Even more intriguing are the findings that patterns of epigenetic modification can be changed in response to environmental conditions. And there's some evidence that epigenetic changes due to environment may be inherited at least for a few generations. Now let's consider honeybees, where one larva is raised to be a queen, while its sisters with identical DNA grow up to be sterile workers. It has been long known that larvae destined to be queens are fed a different diet than workers. Would-be queens are fed a concoction called royal jelly that mysteriously turns them into fertile queens. We now know that royal jelly contains compounds that affect epigenetic modifications. One of these, phenylbutyrate, inhibits the enzymes that take acetyl groups off the tails of histones. For the queen, the acetyl groups remain on the histones, the chromatin is more open, and genes in the region for becoming a queen can be expressed. For worker bees, the enzymes that take acetyl groups off the tails of histones are not inhibited. So those queen maker genes are not expressed, and those bees develop differently than the queen. There also seem to be differences in the methylation patterns in the DNA. Future queens lack the methylation seen in the DNA of bees that develop into workers. In fact, if the enzyme that adds the methyl groups to DNA is intentionally inhibited in larvae, ordinary sisters turn into queens, royal jelly or no royal jelly. So could our diet or other environmental factors change our epigenetic marks and our fates, or even the fate of our children? Perhaps. It's clear that epigenetic modifications can change with an individual's experience and exposure. Researchers are working hard to determine how far-reaching the effects of these changes can be. So far, we've only looked at one stage of gene expression, transcription. There are also other steps in gene expression at which cells can regulate what proteins get made in cells. We'll take a look next at some post-transcriptional mechanisms that change protein profiles in cells. Eukaryotic messenger RNAs, as you know, must be spliced to remove introns, those non-coding regions within the protein coding information in the transcript, and join the exons to make the continuous coding sequence. But as we saw earlier, splicing does not always use all of the exons in a transcript. Instead, different combinations of the exons may be joined together to make different mature messenger RNAs that will then direct the synthesis of different proteins. This is called alternative splicing, and what it means is that transcription of the same gene could result in the production of different proteins in different cells, or even in the same cell at different times. But there are even more possibilities for regulating and altering the expression of proteins. And strangely enough, one major way that the expression of a gene may be regulated is by a group of RNAs called small regulatory RNAs. Yes, you heard that right. We've been talking about messenger RNAs all this time, the bearers of information for making proteins. And now we're going to see how the amount available of these messenger RNAs and whether they are translated is regulated by a whole other set of RNAs known as regulatory RNAs. Now, until the late 1990s, biochemists knew nothing about regulatory RNAs or the role they played in gene expression. More recently, they've been explored as possible treatments for everything from cancer to viral infections. Now, we'll touch on two kinds of regulatory RNAs, about which the most is known so far: microRNAs and short interfering RNAs. The microRNAs are Aunt Millie, who's got a million things on her mind. The short interfering RNAs are Uncle Simon, who's a simple fellow, usually focused on just one thing. Both are small, non-coding RNAs, and they silence genes by base pairing with target messenger RNAs and marking them for degradation or by blocking their translation. This sort of regulation is called RNA interference, or RNAi. Mature microRNAs and silencing RNAs team up with a class of proteins called Argonaute proteins to form structures called RNA-induced silencing complexes, or RISCs. The regulatory RNA provides a short single-stranded guide RNA, roughly 20 bases long, and that together with the Argonaute protein is the functional silencing complex. The combination is deadly. The guide RNA picks out the messenger RNA target, and the Argonaute is the hitman. The guide RNA and the silencing complex first base pairs with its target messenger RNA. For complexes containing silencing RNAs, this triggers the degradation of the messenger RNA by the Argonaute protein. RISCs containing microRNAs may either degrade the messenger RNA or simply block translation by preventing the ribosome from acting. In either case, the protein encoded by the messenger RNA will not be made. MicroRNAs tend to be less specific than silencing RNAs, and because of this relaxed specificity, Aunt Millie can silence many different messenger RNAs. Silencing RNAs must match perfectly with their messenger RNA targets. Simple Simon can only interfere with one specific messenger RNA. Now, there's still a lot to learn about these regulatory RNAs, and there may be more surprises in store. The really exciting thing about RNA interference, though, is that we can synthetically make RNAs that could be coupled with Argonaute to target any messenger RNA we want. Laboratory studies have shown that RNA interference can successfully be used against viruses like HIV and human papillomavirus. RNA interference can also be used to target specific cancer-related genes in melanoma, leukemia, and pancreatic carcinomas. A major challenge in moving laboratory findings into clinical use is that it is difficult to deliver the silencing RNAs to the cells in the body where they are to act. Despite this, the first ever silencing RNA therapy was FDA approved in late 2018 as a treatment for a genetic disorder called transthyretin amyloidosis. This disease involves an abnormal buildup of amyloid proteins. It's caused by a mutation in one copy of the gene for a protein called transthyretin that is made in the liver. Because lipid complexes are naturally taken up by the liver, the scientists were able to deliver the silencing RNA packaged in lipid nanoparticles. The nanoparticles delivered the silencing RNA into the liver cells where it successfully turned off the expression of the mutant transthyretin gene. Now, we've been discussing ways in which gene expression can be regulated after transcription. But what about translation? To no one's surprise, cells have clever ways to regulate gene expression at this point too. We're going to take a look at one such mechanism that regulates the production of two proteins we need for the uptake and storage of iron in our cells: transferrin receptor and ferritin. The first of these is a membrane protein called transferrin receptor, and it's involved in importing iron atoms into cells. Now, as you know, iron is a crucial component in many essential proteins like hemoglobin and cytochromes. The second protein needed to handle iron, known as ferritin, is good at packing iron atoms away in a ball-like structure within cells. Now, this is important because free iron atoms floating around in the cell can create dangerous free radicals. When levels of iron in a cell are low, the cell needs to take up more iron and needs more of the transferrin receptor. And when iron is abundant in the cell, more ferritin is needed to safely store the iron. Now, interestingly, the expression of both these proteins is regulated by a remote sequence in the untranslated regions, or UTRs, of their messenger RNAs. This regulating sequence, called the iron response element, or IRE, folds on itself to make a hairpin structure. The IRE can be bound by a binding protein. Okay, now watch how the IRE and its binding protein respond to the levels of iron and control the production of transferrin receptor and ferritin. The messenger RNA encoding the transferrin receptor has several IRE sequences in the 3' UTR of the transcript that can be bound by the IRE binding protein. When iron levels in the cell are high, there's no need to make more transferrin receptor. Under these conditions, the iron binds the IRE binding protein, which then undergoes a conformational change and it dissociates from the IRE. This leaves the 3' UTR susceptible to attack by RNases, leading to degradation of the transferrin receptor messenger RNA. But when iron levels are low, the IRE binding protein remains bound to the 3' UTR of the messenger RNA, stabilizing it and permitting more transferrin receptor to be made. Okay, what about the ferritin? Well, remember that we need more ferritin when iron levels in the cell are high. And in the ferritin messenger RNA, the IRE is in the 5' UTR. When iron levels are low, the IRE is bound by the IRE binding protein, blocking translation of the ferritin messenger RNA. And if iron levels are high, then iron binds to the IRE binding protein, which dissociates from the IRE. This frees up the 5' end of the ferritin messenger RNA for ribosome assembly and translation, producing more ferritin. Because too much iron absorption is so harmful, we can be glad that the body has gene expression helping to regulate iron uptake. So cells regulate which proteins are made, how much of each protein is made, when they're made, and how long they're made. These, in turn, determine our traits and what our cells can do. Now, there are 3 common themes here. First, the binding of proteins to specific DNA sequences can increase or decrease transcription, as we saw in the lac operon. Second, binding of proteins to specific sequences in RNA can control translation, as with ferritin and the transferrin receptor. And a third theme is that even if an RNA is made, its levels can be controlled after the fact. Regulatory RNAs can target particular messenger RNAs for destruction or block their translation. So we've seen that what we inherit and pass on are not just our DNA sequences, but also some of the controls on expression of those sequences through epigenetics. Perhaps most curiously of all, the epigenetic inheritance lasts only a generation or 2, whereas the DNA is forever, or at least until mutation occurs.